Molecular basis of inheritance, part 6. In this video, you will be learning about translation and how proteins are being formed. Now, before that, let us try to understand tRNA as an adapter molecule. tRNA is also known as soluble RNA or sRNA. Is synthesized in the nucleus by the DNA and later transported to cytoplasm. Many tRNAs are synthesized in the laboratory. tRNA's shape resembles that of a clover leaf and hence it is also called clover leaf model of tRNA. It has an amino acid binding site to which an amino acid is attached. It also has an anticodon arm with a recognition site provided with a nucleotide triplet which is complementary to a particular triplet codon in the mRNA to which it would get linked during protein synthesis. It also has a small extra arm. tRNA acts as conveyors to bring appropriate amino acids to the ribosomes as per the code in the mRNA for protein synthesis. Translation involves steps resulting in the actual biosynthesis of protein as per the nucleoside sequence of mRNA. Now let us look at the steps in detail. Attachment of mRNA to 30S ribosomes. The mRNA coming from the nucleus attaches to the 30S ribosome in the presence of F factor. The mRNA is provided with a series of triplets, that is 3 nucleotides, and each triplet forms a code. The next step is activation of amino acids. 20 different kinds of amino acids are present in the cytoplasm in the amino acid pool. These amino acids need to be activated before being transferred to tRNA. Enzyme amino acyl synthetase and ATP will activate each amino acid in the presence of magnesium ions to form amino acyl adenylate enzyme and pyrophosphate. Since there are 20 different amino acids, there should be 20 different amino acyl synthetase for the activation of 20 different amino acids. Now, the next step is transfer of activated amino acids to tRNA. When the amino acyl adenylate enzyme complex comes in contact with a specific tRNA, the amino acyl adenylate enzyme complex is recognized by amino acid binding site to which it is attached. The enzyme then gets separated. Now the initiation of protein synthesis. Now the amino acid tRNA with the activated amino acid at its binding site moves towards the mRNA in the 30S ribosome and attaches to the first triplet AUG of mRNA to form the initiation complex. And this takes place in the presence of guanosine triphosphate and IF1, IF2 and IF3. These are the initiation factors. Chain initiation codon. AUG of mRNA is the codon for methionine and it is called chain initiation codon. It is found that AUG for methionine is always the initiator codon in protein synthesis. Immediately after the formation of initiation complex, the 50S ribosomal subunit moves on to 30S ribosome and the two subunits unite themselves to give rise to 70S ribosome. Initiation of protein synthesis has started and amino acid tRNA complex is at the A site of the 50S ribosome. The A site is the amino acyl tRNA binding site. In prokaryotes, AUG always codes for methionine and hence AA1, that is the first amino acid, is always a methionine. The first codon moves on to the P site, that is peptidyl tRNA site. The amino acid 1 is left at the site and tRNA moves out via exit site. This happens in the presence of GTP. The second codon is in the amino acid site and AA2 tRNA complex with the second amino acid is attached to this code. Now this one will move to the P site and AA3 comes to the A site and tRNA moves out of the exit site. So like this, if you look at the amino acids keep coming and keep moving on as though they are moving on a conveyor belt. As a result, the polypeptide chain gets synthesized by addition of correct amino acid and the whole process will go on rapidly. Termination and release of polypeptide chain is accomplished by the presence of one of the terminating codons which could be UAA, UGA or UAG. the polypeptide chain is simultaneously released. UAA, UGA and UAG are nonsense codons as they do not code for any amino acid. Once when these codons come, then the synthesis of polypeptide chain stops. When the polypeptide chain is released, the first amino acid methionine is removed from the chain by deformylase. Any amino acid from the end of the polypeptide chain that is not needed is also removed by exopeptidase. Finally, the polypeptide chain with the correct amino acid sequence fold itself to form the protein or the enzyme.